Insulators and boundary elements as regulatory sequences in transcription in eukaryotes. When one tries to understand insulators and boundary elements, it is very clear that there is a very thin line of difference between insulators and boundary elements in terms of the functioning. So, in fact, boundary elements are also called as insulators per se. So, let us look at the learning outcomes of this session. Insulators and boundary elements are, of course, cis-acting elements that are able to differentiate chromosomes into topologically different domains, thereby protecting and expressing gene from its surrounding environment. So we'll come to understand this as we look at different examples. Insulators are able to block enhancer activity on a promoter. This is especially depending on how the insulators are positioned. If they're positioned between an enhancer and a specific promoter, then the enhancer activity on that specific promoter can be blocked. Insulators can also act as barriers or boundaries, and they can protect a gene by forming what is called as boundaries around that gene. They are barriers to advancing condensed chromatin. So we all know that when you have highly condensed regions in the chromatin, they are called as heterochromatin. And if the heterochromatin goes and tries to condense the euchromatin region, any unique gene present in the euchromatin region will now get silenced because it is getting converted into heterochromatin. So this, chromatin, heterochromatin, making a euchromatin more heterochromatin is prevented by the presence of these boundary elements. So the insulators can be sequences from anywhere between 300 base pairs to 2 kb in length, and many of the cis-acting elements are present as short stretches, but they are short stretches which are distributed over a range or over a range of base pairs possibly. So you have as small as 300 base pairs to as long as 2 kbs in length. Now let us understand insulators by looking at certain examples. So one of the best studied example of the influence of how a cis-acting element is working as an insulator is in the chicken beta globin gene. It has been also seen that a similar effect is seen in the human beta globin gene. But here we are taking basically the example of the chicken beta globin gene. So let us look at the locus of the beta globin gene and understand how it is that the insulators are working towards the beta globin gene expressions. So when you look at the beta globin genes per se in the chicken, you will find that this is the locus, and this is what is called as a region that has all the beta globin genes. Okay, so we know that you have several different subtypes or several genes present that finally will comprise the beta globin protein. Now, interestingly, what is observed is on either side of the locus of the beta globin genes is present insulators. So you have over here what is called as the HS4. This is towards the 5' end of the beta globin gene locus, and you have 3' HS on the 3' end of the beta globin locus. This is called as HS4 because of the fact that this is DNA's one hypersensitive region, and from that the name comes HS4 or 3' HS. You know that you have enhancers present, okay, and you can see that here there is an enhancer that is present, and what you can also see is that just upstream of the insulator region is present a highly condensed region of chromatin. So the methylated nucleosomes enable it to form a highly condensed part. So this is becoming heterochromatin. And this over here is what is called as locus control region of globin, and this acts as an enhancer. So this is basically stimulating the synthesis or transcription of the beta globin genes from the promoter region present over here. Now, because you have these insulators, you can see that the folate receptor gene is topologically differentiated from the beta globin genes, which in fact are further topologically differentiated from the odorant receptor gene. So you can say this is one topologically different region, this is another topologically different region, and this is another topologically different region. Why is this so? This is thanks to the insulator regions. So therefore, this is a clear indication as to how the insulator is creating topologically differentiated domains. And within the domain, you can have a cluster of genes or you can have a locus. So in fact, what is also observed is that the heterochromatin is also responsible for differentiating two different gene loci. So therefore, this is also what is called as a uh, as an important factor that can differentiate two things. But then what happens is if it goes more and more differentiated. So if this heterochromatin base becomes more and more condensed and moves towards this LCR region, then what is going to happen? Even this portion, that is the globin portion, will become heterochromatin. Then what is going to happen is that the beta globin genes are going to be silenced because of this region becoming heterochromatin. So the presence of this insulator over here is preventing the chromatin region from getting into the euchromatin portion, thereby protecting this locus from the heterochromatin. So this is an extremely important factor that is associated with the insulators. Now, this enhancer which is present is able to, okay, before we get into that, let me also make it clear that these globin genes that are topologically differentiated are late genes. That means they are transcribed late in the development, okay, of the chicken. While the folate receptor genes are expressed much early in development, okay, so they are called as early genes. So you can already see that because they are topologically differentiated, one can be transcribed early while the other can be transcribed late. And also what has been observed is because of the presence of the HS4 component, this enhancer, okay, that is there is able to influence or stimulate the expression from this promoter. So it is able to highly express the folate receptor gene, okay, but it is not able to express or it is not going to have any effect on the beta globin genes because of the presence of the insulator. So in this case, the HS4 is acting both as an insulator because it is blocking the activity of the enhancer, and it is also acting as a boundary element because it is preventing the condensed chromatin from making the euchromatin region condensed. So it is acting as a boundary element and it is also acting as an insulator per se, whereas the 3' HS is acting only as an insulator. Why is it acting as an insulator? Because the LCR will not able to influence the promoter of this odorant receptor gene. It will have its own enhancer which is able to stimulate the expression of this. Whereas that enhancer that is present over here will not be able to influence the expression of the beta globin genes. So therefore, this acts as an insulator for any enhancer present downstream, and this insulator is acting as a enhancer blocking region for the enhancer that is present upstream. So you can also see how the beta globin genes are not influenced by the enhancer that is present elsewhere. Okay, so that is something that has been very categorically understood with respect to the functioning of the insulators. They are not, or they are protecting the genes from getting influenced by signals from surrounding signals. Okay, so this is one example. Let us look at another example. You have the human TCR, that is a T cell receptor alpha delta locus. So this comprises the genes that are responsible for forming the T cell receptor, and we know that there are two subunits. It can also it is primarily the alpha delta subunits that make the T cell receptor. So let us look at the construct of the locus of the human TCR. So you can see that you have a promoter over here that expresses or is responsible for expression of the delta genes, while you have another promoter that is responsible for transcription of the alpha genes. And one can also observe that there is an enhancer for the alpha genes and there is an enhancer for the delta genes. And within the two is present what is called as the blocking element alpha delta. This is basically the insulator region. And this insulator region is ensuring two things. One is that this enhancer, which is specifically for the delta genes, can stimulate transcription of from the promoter for the delta genes, while it is not able to influence, because of the presence of the blocking element, the expression of the alpha promoter regions. Same is the case with the enhancer for the alpha genes. So this is able to definitely stimulate transcription from the promoter region for the alpha genes, but it is not able to influence the transcription from the delta promoter, which means that this enhancer is not having an effect on these genes, while this enhancer is not having an effect on these genes. Why? Because of the presence of the blocking element in between. This is a clear indication of the fact that the bead element is displaying what is called as position dependent enhancer blocking activity, which means because it is present between an active enhancer and a promoter, right? Or an active enhancer and another promoter, okay? It is blocking the activity of one of the enhancers with respect to the other promoter. So if this were present here, then it would affect this promoter too, correct? If it were present here, then this would affect the activity of this enhancer as well. So depending on where it is positioned, it is likely to block the activity of the enhancer that way. So therefore, this is very clearly displaying that there is what is called as position dependent enhancer blocking activity. Let us look at another example. This example is that of the xenopus ribosomal RNA repeats. So in this locus, what has been found is that there are the 40S genes, and we know that 40S is synthesized as one transcript, but then it is eventually processed into 18S, 5.8S, and 28S. And you have several clusters of this that is present, separated by regions which are called as repeat organizers. So here what is present is a cluster of cis acting elements which function as insulators, and therefore they are called as repeat organizers. And you will find that every locus for the 40S gene is being separated or differentiated topologically by the RO region. Now, very clear indication that you again see what is called as a polarized enhancer blocking. Now, this decides basically the polarity of the action of enhancer. So this enhancer is able to stimulate the transcription of these genes, whereas this enhancer is able to stimulate the expression of these genes. You cannot have this regulating or influencing the transcription of these. Neither can these be influencing this. So you do not have, so you can see that in only one direction, you have the enhancer stimulating transcription of the genes downstream of it. So this is very clear indication again of what is called as position dependent um, enhancer blocking, and also deciding the polarity of the functioning of the uh, insulator or the enhancer per se. So this is very important finding again. So from all of these, you kind of get an overview of how the insulators are functioning. Now, there were a lot of experiments that were carried out in vitro, okay, to actually characterize enhancer blocking. And we will just take a few examples of studies that were carried out to look at the enhancer blocking of by the insulators. So, transgenic constructs were made with two promoters and two enhancers. Okay, so let us look at what the transgenic constructs here. So you can see over here that there are two promoters, okay, transcribe in two different directions, okay, and you have enhancer 1 and you have enhancer 2. Now, what is interesting to note is that in one cell type, the enhancer 1 functions, and in the another cell type, the enhancer 2 functions. So we already know that enhancers can have tissue specific influence or tissue specific transcription regulation. So therefore, this is very clear that this enhancer can function in this cell type and this enhancer can function in this cell type. Now, within the transgenic constructs, one of the transgenic constructs was made in such a way that an insulator cis-acting element was added between one proactive enhancer region and a promoter. So when you don't have an insulator, this enhancer is able to stimulate transcription from both the promoters, right? But when you have the insulator added, okay, then what is observed is that this insulator blocks the enhancer activity for this promoter. So you can very clearly understand that positioning the insulator between the enhancer and the promoter has resulted in decreased enhancer promoter interaction, thereby it affecting the transcription from one of the promoters, but the other one is not affected. So the positioning of the cis-acting insulator is not affecting the other promoter regions. It is only affecting the promoter that is present or that it is present between the enhancer and the promoter per se. Now, when you look at the cell type 2 as well, in the cell type 2, you have this enhancer working. So, when you have no insulator cis-acting element present, then the enhancer is able to activate both. So, they are able to stimulate transcription from both the promoters. But when you have an insulator positioned between the enhancer and one of the promoters, okay, then you can see how it is able to stimulate transcription from this promoter because there is no insulation here, but it is not able to stimulate transcription from this promoter because of the presence of the insulator. So effectively what you are trying to understand is, what we are trying to look at is that the insulator functions when it is positioned between an active enhancer and a promoter. Okay, this is very clear. Now let us look at another study that was carried out, and this study was carried out in such a way that a compound element was constructed. What does it mean by a compound element? It means that you have a gene construct which has both an enhancer and an insulator cis-acting region. Now, in their study, what was done is two constructs were made such that this comprises orientation 1. So, in this case, you have the insulator present between the enhancer and the promoter region, and in the other orientation you have the insulator present within the construct, that it is proximally present to the enhancer region, but it is not between the promoter region and the enhancer. So the effect is also very clear when the enhancer is present such that there is an insulator region between it's the promoter that it is acting on, okay, then this enhancer is not able to stimulate transcription from here, so which means that this insulator is blocking the enhancer. But when the orientation is such that between the enhancer and the promoter you don't have an insulator region, even if the insulator region is present, it is not able to block the enhancer activity. So this is again an example of how position of the insulator is important to decide whether it can block the enhancer activity or not. Now let us look at another example where the same two orientations were used, but the construct containing the enhancer and the insulator was inserted between an already existing enhancer and a promoter. So the effect is also very clear. When you have the insulator present between the enhancers and the promoter, you can see that the activity of both the enhancers are blocked. But in this orientation, you do not have the blocking of this enhancer, but you definitely have the blocking of this enhancer. So this is something also that is suggestive of the fact that there is what is called as a polar enhancer blocking. So only one orientation of the enhancer insulator is blocking the enhancement, the other one is not. So this is again important, or this is just basically understanding how insulators are able to block enhancers. Now, of course, what is understood is that since they are cis-acting elements, they would definitely have trans-acting elements that would bind to that, and and therefore, based on the binding of these proteins to the insulators, you will have an effect on the transcription. So there are many studies carried out, and there have been many DNA binding proteins found that are able to interact with the blocking elements or the insulators. So one of the examples is the CCCTC binding factor, that is the CTCF. This has been majorly found in the vertebrates. While a lot of studies having been done in Drosophila, it has been found that there are several different DNA binding proteins that can bind to different blocking elements or insulators. So you have what is called as the SUWH proteins, you have the BEF proteins, you have the ZW proteins, you have the GAGA factor. All these are different repressor proteins that are influencing transcription. And these have all been found in the Drosophila melanogaster. So we will look at one or two examples and try to understand how the binding of the protein counterpart to the blocking element influences transcription or influences the activity of the insulator itself. So let us take the example of the SUHW repressor protein. Now this SUHW repressor protein has been found to bind to insulators present on gypsy retrotransposons. So there are retrotransposons named as gypsy, and within the gypsy retrotransposons are present the boundary elements or the cis-acting elements which function as insulators. To this insulator specifically, the SUHW repressor proteins can go and bind. So, looking at a construct or a gene locus for a promoter that is activating a gene that is going to give rise to yellow pigment. So, this is basically the yellow pigment locus of the Drosophila. And this has basically three enhancer regions, okay? And what has been done is in between the enhancers, the gypsy retrotransposon is inserted. So because the gypsy retrotransporter is inserted at this position, the SU protein will come and bind. Now when the SU protein comes and binds to this retrotransposon, which has the cis-acting elements or which has the insulators, what was observed is that expressions from all the enhancers were silenced by the SU. So effectively, this insulator is blocking enhancer activity from all the enhancers. Okay, but when this SU is bound to by another protein called the MOD protein, what was observed is that because of the binding of the MOD to the SU and this SU binding to the insulator region, it is blocking the enhancer activity of these two enhancers, but it is not blocking the enhancer activity or of this enhancer. So effectively, again, a positional enhancer blocking has been carried out by the proteins that are binding to the insulator regions. So therefore, this is an example which is saying that the cis active elements are not working in isolation. They are being, the functionalities are being modified based on what is the protein that is coming and binding to these insulator regions. So when you have this enhancer activating, you will have the yellow product form. But when you have just the SU, you will not have the yellow product being formed. So the difference is in what? The difference is in what is the proteins that is associated with the insulator region. So therefore the protein counterparts are also having a very important role to play. Now let us look at one another example with respect to the protein counterparts that bind to the blocking elements. So let's consider the mouse IGF2 H19 locus. Now, the mouse IGF2 H19 locus, when you look at the two homologous chromosomes, one finds that one is definitely from the maternal side, and so therefore you have a maternal allele and you have a paternal allele. Now, interestingly, what is found is in the mouse IGF2 H19 locus, between the IGF2 and the H19 promoter is present four CTCF binding regions, that is CTCF binding. So there are four cis-acting elements which are present. In humans, it has been found to have seven such regions, but in this is in mice and there are four. So these are four sequences to which the CTCF can come and bind. As soon as the CTCF comes and binds to the insulator regions, okay, what is observed is this enhancer, which is present downstream of the H19, is able to stimulate the H19 expression, but it is no longer able to stimulate the IGF2. So in case of the maternal allele, the IGF2 is not functional. In case of the paternal allele, what has been observed is because of imprinting, genomic imprinting, the cis-acting elements, that is the boundary elements or the blocking elements, have been methylated. Because they are methylated, what happens is that the CTCF factor, the protein, is not able to come and bind to these regions, and thereby it is not able to function primarily as an insulator. And so the enhancer, which is downstream, is able to initiate or stimulate transcription from both the H19 promoter as well as the IGF2. So in case of the paternal allele, both IGF2 and the H19 are functional, whereas in the case of the maternal allele, only the H19 is functional, the IGF2 is blocked. So therefore, the CTCF binding is essential for blocking the enhancer activity. So you can see that it is not just the cis-acting elements, but the protein counterparts that also influence the transcription. And so another example of how the protein counterparts are influencing the transcription. One another observation with respect to insulators and the protein counterparts is that they are able to form very specialized chromatin organization. So let's say, for example, we all know that in the nucleus is present within, that means in the nucleus, within the nucleus is present the nucleoplasm, and within the nucleoplasm is present what is called as the matrix. So you have a nuclear matrix present. And it has been observed that many parts of the DNA, many segments of the DNA, many sequences in the DNA are able to attach to the matrix. So those regions are what is called as the matrix attached regions. And interestingly, the matrix attached regions, that is the segments to which you have the proteins binding, and the proteins in turn are binding to the nuclear matrix. One clear observation is that the binding of these proteins to the matrix associated or attachment regions are allowing or enabling the chromatin to form separate loops. So you can see that topologically you have genes present over here, differentiated from genes present over here, differentiated from genes present in this loop. So you can see that special chromatin organization is possible because of the interaction of the protein with the DNA, and that protein again with the nuclear matrix. And when that happens, you have these specialized loops being formed, clearly indicative of that genes that are present over here may be regulated differently from the genes that are present here. So it has been observed that if there is an enhancer present in this loop and a promoter present in another loop, then this enhancer will not be able to stimulate transcription from this promoter. Why? Because of the presence of this insulator or the presence of these blocking elements. Okay. What has been also found is that within a loop, you can have subloops being formed, because again, you can have certain insulator regions bound to proteins, and because of the binding of these proteins, you can see that subloops are formed. Now, suppose you have an enhancer region over here and you have a promoter region in another subloop present, again, you will find that this enhancer cannot stimulate transcription from this promoter. So the organization of chromatin into loops is also differentiating the gene loci topologically, and thereby protecting these genes from getting activated by surrounding environment or surrounding signals. So this is something that has been very clearly observed with the insulators and the boundary elements. Now, interestingly, there is another thing that has been observed, which is called as insulator bodies. So, if you can see over here, when you have gypsy retrotransposons present in different parts of the DNA or different parts of the chromatin, those parts of the chromatin can be brought together by what is called as an insulator body. How is that formed? Every retrotransposon that has cis-acting elements can bind to the SUHW, for example, right? These SUHW can in turn bind to MOD, and these MOD can cooperatively bind to another protein, forming what is called as an insulator body. So you have loops, chromatin loops present all around the insulator body, and the insulator body is able to influence the transcription regulation from different promoters that are present in these loops. So if you have an enhancer present here, it is able to stimulate transcription of promoter 1, but it cannot. So the insulator body is actually blocking the enhancer activity of this E1 for any of these promoters that are present in the other loops, including in the same loop where you have the P2. Now, if you had an enhancer present over here for P2, again that enhancer would only be able to stimulate transcription for P2, but not for the other promoters that are present in other loops, thanks to the presence of the insulator body. So you can say that the insulator body is again able to position dependent influence what is called as position dependent enhancer blocking, and also deciding the polarity of the enhancer activity. So this is again an interesting phenomenon that has been observed. Another interesting concept is that of insulator pairing. So what has been observed is, when you have insulators say present between an enhancer and a promoter, and say for example the orientation of both the insulators are in the same direction, and these have sequences that can say for example bind to proteins, and the proteins can bind to each other, thereby the insulators can pair up, as you can see over here. Now, the way the insulators pair up is very strategic. We all know that the insulators are able to, you know, create what is called as independent cis regulatory networks. So you have, because of the presence of the insulator, this as a separate, cis network, this as a separate network. So if you have the insulator present between this enhancer and promoter, this enhancer will not be able to stimulate this promoter because the insulator is blocking the enhancer activity. Now, in case the two insulators pair up in a particular orientation and a loop is formed, but a loop is formed in such a way, so you can see that chromatin organization is happening because of insulator pairing. But if due to the loop formation, you have further, okay, the enhancer away or distally present from the promoter and the insulator present between the enhancer and the promoter, then it will block the enhancer activity. But say for example, you have the insulators here, you can see that the orientation of the insulators are different and therefore the pairing of the insulators can be different from this. In such a scenario, the looping will be different. So if the looping of these oriented insulators are done in such a way that you have the promoter region coming close to, so where it was earlier farther away, now it has come close to the enhancer, then what is happening? The enhancer is now able to stimulate the activity of the transcription from this promoter. So in fact, insulator pairing has been in such a way that it is not coming in between the enhancer and the promoter. In turn, it is bringing a promoter close to an enhancer to initiate transcription. So you can see how insulator pairing with regard to its by binding with each other, okay, or with regard to the orientation in which it is binding and therefore thereby influencing the looping of the chromatin, can decide whether an enhancer can be blocked or an enhancer can be actually kept functional. So therefore, this is also an important fact that topology of the loop form depends on the orientation of the insulators and the pairing interactions. And the looping can bypass the blocking if the enhancer and promoter are brought close to each other. An interesting concept. And also looking at the fact that so far we had been looking at the fact that the insulators are able to, only block the enhancer activity. So here too, as an insulator between an enhancer and a promoter, it is blocking. But it may be responsible for bringing a promoter and enhancer close by because of its specific organization, chromatin organization. You may have the insulator pairing bypassing the enhancer blocking as well. And this could depend on different signals. Okay, so let us make the concluding remarks. Insulators as cis elements regulate transcription by differentiating gene loci or genes from each other so that the genes are not activated by any inappropriate signal. They can act as enhancer blockers or they can act by being boundary elements between heterochromatin and euchromatin, ensuring that the heterochromatin do not silence genes close to them. There is an example also very clear about the telomeres being able to, if the telomeres keep condensing, they may be able to actually, silence genes which are very close to the telomeres. So is the case with centromeres. Enhancer blocking by insulators can be decided by the position and orientation of the insulator cis elements, which of course can vary in length, and also the binding of the proteins to these insulators is extremely important. There are specialized organization of chromatin that is aided by the insulators and the associated proteins which are able to insulate promoters from enhancers. So insulators boundary elements are an interesting mix of versatile regulatory elements that have shown to be able to enhance transcription or in certain cases repress transcription of differentially separated gene loci. Thank you.